Prokaryotes lack internal membrane-bound organelles, and eukaryotes came from prokaryotes. So our prokaryotes, they can be rod-shaped, spherical, or spiral-shaped, and they don't have all of these membrane-bound organelles on the inside. Their DNA is one long circular sequence, and they also have free-floating ribosomes. Now, they have regions within there, but there are no actual membrane-bound organelles. Of course, they are surrounded in a outer plasma membrane. They also have a cell wall, which prevents them from bursting if they were in a very watery environment. That cell wall is another way to distinguish one prokaryote from another prokaryote based off of how they respond to stain. If they have a lot of peptidoglycan in their cell wall, they will stain gram positive. If they don't have very much peptidoglycan, they will stain gram negative. Prokaryotes also have a capsule around the outside of the cell wall, which helps prevent them from drying out, allows them to attach to most surfaces, and will also protect them within a host from the immune system of that host. Some prokaryotes are also motile. Eukaryotes have compartments, as we've been talking about. So they have all of these membrane-bound organelles, but the leading theory is that they came from prokaryotes. The original prokaryote that led to eukaryotes was a heterotroph, which means it was an eater. But before we get to that point, the DNA was surrounded as the outer cell membrane enfolds, and we end up with a nucleus. After we have a nucleus, more of that enfolding of the outer cell membrane starts making an endomembrane system, like when we start thinking about endoplasmic reticulum and Golgi apparatus. Again, these are all evolutionary advantages. It's not that the original cell can't continue to exist, but these are successful and they continue to be carried on. That heterotroph, that original prokaryote, also eats other prokaryotes. Typically, when something is brought in through endocytosis, it would be destroyed and the material within it could be recycled to be used by this original cell. However, when this cell engulfs this, instead of destroying it, they live together, and it is a symbiotic relationship where both benefit. This is where the original mitochondria are thought to have come from. One prokaryote engulfing but not destroying another prokaryote. And that's how we assume we got mitochondria within animal cells. Now, again, these are still heterotrophs. When some bacteria can be photosynthetic, like cyanobacteria, which do have thylakoids and can do photosynthesis. When we have this original prokaryote, it engulfs but does not destroy these other bacteria, and we think that is how the chloroplast originated within plant cells. So the claim is that mitochondria and chloroplasts inside eukaryotic cells were once free-living prokaryotes. So what evidence exists? The fact that mitochondria and chloroplasts are about the same size as modern-day bacteria. In addition, mitochondria and chloroplasts have a double membrane. One membrane is from the original prokaryote, and as it went through endocytosis, it gained a second membrane from the host prokaryote. In addition, another form of evidence is that mitochondria and chloroplasts contain DNA, and the DNA of mitochondria and chloroplasts is ring in structure and very similar to prokaryote DNA. In addition, mitochondria and chloroplasts also have ribosomes, which are very similar to the ribosomes of prokaryotes. And while a lot of the proteins that exist within mitochondria and chloroplasts come from the host cell, from the actual eukaryote, mitochondria and chloroplasts do make some of their own proteins. Finally, mitochondria and chloroplasts reproduce more similar to prokaryotes on their own, independently from the host. So they go through a process that's like binary fission, which is like prokaryotes. So again, prokaryotes lack internal membrane-bound organelles, and the endosymbiotic theory says that many membrane-bound organelles of eukaryotes came from originally prokaryotes.